We can start by surveying the action potential graph of a neuron as a whole and then break down each step in more detail. We start at resting membrane potential, which is around minus 70 millivolts. In response to a stimulus, the membrane potential becomes less negative or depolarized and approaches zero. During depolarization, sodium rushes into the cell from the outside, thereby increasing the membrane potential. As more sodium enters the cell and the membrane potential becomes more positive, overshoot occurs to about 40 millivolts. Once the action potential begins, the membrane potential turns the corner and decreases again. We call this repolarization, and it occurs due to potassium channels opening and sodium channels closing. In other words, positively charged potassium is leaving the cell, and there's no more positively charged sodium entering the cell. This actually happens so dramatically that there's a slight overshoot known as hyperpolarization, where the membrane potential is actually lower than the resting minus 70 millivolts. After all of this, old faithful sodium-potassium ATPase regenerates the ionic gradient that restores the membrane potential to minus 70 millivolts. And all that together is an action potential. Now that we've established these foundational concepts, let's look a little deeper at how a signal is transmitted. First, after an action potential, neurotransmitters are released from one neuron into the synaptic cleft, which is the very small space between the terminal of one neuron and the dendrites of the next. On the plasma membrane of the dendrites, there are a ton of different receptors, each specific for a different type of neurotransmitter. When a neurotransmitter binds to an appropriate receptor, that receptor will allow a specific ion to flow inside. This ion flow slightly changes the membrane potential, an effect called the graded potential. Most graded potentials are excitatory, meaning that they raise the membrane potential, making it more positive. Let's keep putting the pieces together. We have an excitatory potential at a postsynaptic membrane, so we can call it an excitatory postsynaptic potential, or EPSP for short. However, inhibitory graded potentials, or IPSPs, are also possible. These make the membrane more negative, resulting in hyperpolarization. Graded potentials varying in size can add up together if they are located close to each other or happen within a very short time span, phenomena that are technically referred to as spatial summation and temporal summation respectively. Graded potentials can also propagate a short distance along the soma and towards the axon hillock. If enough of these graded potentials add up in that short time span, we will get an action potential. But since action potentials are the stars of the show here, we are going to save diving into the details of this mechanism for the next video, so stay tuned.